Good morning, students. Good evening, students. I am Dr. Stefan, and today we are continuing with our Biomolecules 2 video. And today's class will have to deal with amino acids and proteins and nucleic acids. So we are going, as we pass through the class, I expect each and everyone to have basic knowledge as I emphasize on some particular points. So let's start. So first of all, what are amino acids? So amino acids, they are macromolecules that are made up of carbon, hydrogen, oxygen, and nitrogen atoms. So they are the building blocks of proteins. So the monomers that make up proteins are amino acids, and they have a general formula. That is, you have a central carbon atom that is linked to a hydrogen group that was not represented here, and at the same time an amino group, a carboxylic group, and an alkyl group. So this R group is a group that is variable. The other groups are constant groups. Okay, so it is very important for you to note that amino acid possess both an acidic and a basic group. So what is the acidic group? The acidic group is the carboxylic acid functional group. The basic group is the amino group. Okay, so this basic group will find itself in solution having a positive charge. This acidic group will find itself having a negative charge. So in normal circumstances in solution, we always observe these amino acids that are found with these two charges that neutralize themselves, so a positive charge and a negative charge, and which we call a zwitterion ion. Okay, so we call amino acid a zwitterion ion, so they exist in solution as zwitterion ions, and we have mainly 20 amino acids in nature. So there are some particular amino acids which animals are unable to synthesize and must therefore obtain these amino acids from plants. So these amino acids are called essential amino acids. Okay, they are essential. Why? Because our bodies cannot synthesize these amino acids, so the only way we can get them is through our diet. So those other amino acids that can be synthesized endogenously mean in our body, are called non-essential amino acids. So some amino acids occur after they have been incorporated into a protein, and they have no DNA code for their synthesis. So example of, we usually call them rare amino acids, and an example is hydroxyproline. So these amino acids, they are not normally found like that in our bodies. So it is only after they are being incorporated in a protein like proline, after proline is being incorporated in a protein, that it is now being converted into hydroxyproline. Okay, so these amino acids that do not occur naturally in our body are called rare amino acids. Then, when two or more amino acids combine, there is a formation of another macromolecule that we call a peptide. Okay, and this combination reaction is a condensation reaction since water is produced. So remember, we saw what a condensation reaction was. So the bond between two amino acids is called a peptide bond. So remember, we said after that condensation reaction there was always a particular bond that links the two monomer units together, and that bond in carbohydrates we called it a glycosidic bond. In lipid we called the bond an ester bond. We called it an ester bond. And in proteins we are going to call that bond a peptide bond. Okay, and the resultant molecule is called a dipeptide, so resulting from the condensation of two amino acid units. So peptide bonds are formed between carboxyl groups and amino groups of two adjacent amino acids. So as a result, there is always a free carboxyl or an amino end for further combination with other peptides. So if many more amino acids combine, we call it, with a dipeptide, we are going to call it a polypeptide, meaning we have a lot of monomers unit. We have a lot of monomer units, so we call it a polypeptide because it has a lot of amino acid. So now I'm talking of proteins themselves. So proteins, they are the most abundant molecules found in the cells, and they form over 50% of the total dry mass of our body. They are essential components of the diet, and they can be converted to both fat and carbohydrate by the cells. Their diversity play a key role in the structural and metabolic activities within the organism. They are constituted by over 100 or more amino acid residues. So a protein is one that is made up of a lot of amino acid residues. Okay, and proteins all have 3D conformation, and based on this conformation, there are some 4 levels of organizations that we are going to see. So we are going to organize proteins, some level of conformation of this protein. We are going to organize it as, firstly, the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. So what is the primary structure? So the primary structure is simply a long amino acid sequence that linked by peptide bonds and disulfide bridges. Okay, and it is the basic level of organization of proteins. So this primary structure just has to deal with the series, or you can also talk of the arrangement of the amino acid or the amino acid sequence that are found on the protein. Then the next is the secondary structure. So we have 2 main types of secondary structures. We have the alpha helix and we have the beta pleated sheet. So the alpha helix is a structure in which you have 2 polypeptide structures that bind around each other and they form a helix. Okay, so wherein the entire structure is in the shape of an extended spiral ring held by hydrogen bonds formed between neighboring CO and NH groups. So we have these polypeptides due to the interaction between the carbonyl groups and the amine groups. We are going to have the polypeptides that are going to wind around each other, forming a kind of helix, okay, which we call the alpha helix. So this is one of the secondary structures of proteins. The next is the beta pleated sheet. So in the beta pleated sheet, you have polypeptide chains that are arranged in a parallel fashion. They either run in the same direction or in opposite directions, right? And the interaction that existing between them are still the interactions in between neighboring carbonyl groups and NH groups. The next type of structure we will see are the tertiary structure. So here in the tertiary structure, the polypeptide groups form 3-dimensional web linkage. Okay, that forms a kind of a globular shaped protein. So you have the polypeptide that is like a straight line that is going to fold on itself and produce a molecule with a 3D conformation. Then lastly, we have the quaternary structure. The quaternary structure, which is simply made up of different polypeptide chains, okay, that are held together by more complex hydrophobic hydrogen or ionic bonds. Okay, so that is the quaternary structure. So you must know that the quaternary structure consists of at least 2 polypeptide chains. Okay, the tertiary structure consists of just a single polypeptide chain. So for example, here we have a diagram that shows to us the primary structure of a peptide or of a polypeptide. So we have, for example, here our different amino acids. So we can observe that these amino acids, they have a central carbon atom, they have an H group, they have an R group that is variable, they have a carboxylic acid group and they have an amino group. Okay, we say that the carboxyl group, the amino group has a positive charge and the carboxyl has the negative charge. The amino has a positive, the carboxyl has the negative. Okay, so here you can observe that in between the OH of this carboxylic acid here has merged with the H of this amine group to form this bond here that we are going to describe and call it a peptide bond. Okay, that link two amino acid together. Same thing here, we have the OH of this carboxylic acid functional group and the H of this amino group that bind together to form the peptide bond that links these two amino acids together. So in between all of these amino acids we have peptide bonds, and we also see that their free ends are always constituted of unbound functional groups, amino functional group and carboxyl functional group. So we call this, because it has the amino functional group, the amino end, and we call the other end the carboxyl end. Okay, so that permits us to have some kind of sense of direction when reading the amino acid. So we say the primary structure is a sequence of amino acids in a protein and is like the order of letters in a word. The primary structure is determined by inherited genetic information. So the primary structure is determined by the DNA sequence or the RNA sequence. Okay, RNA base sequence, the sequence of bases that are found in our RNA. They are the one that code for the primary structure of our amino acids. So for example, here you can observe some different amino acids that are bound together to form a polypeptide chain. Okay, so now the secondary structure, remember we said you have a polypeptide chain that winds together due to interaction between the carbonyl groups and the hydroxyl groups. So you should know that we always, on this amino acid, you always observe that there are some carbonyl functional group, okay, carbonyl functional group and NH functional group. Okay, so the NH functional group of an amino acid can interact with the carbonyl functional group of the other amino acid, and when there is this interaction they form the secondary structure, which can either be an alpha helix or a beta pleated sheet. The beta pleated sheet, we see that you have these parts of the polypeptide chain that runs parallel. They are either parallel or anti-parallel. Then when this polypeptide chain with its secondary structure winds on itself to form a structure that has a 3D conformation, we call that structure a tertiary structure. Okay, we call it the tertiary structure. So you can observe in the tertiary structure that we have a single polypeptide chain that has and beta pleated sheet. So these are beta pleated sheets here indicated by arrows. Then alpha helices. So here there is one alpha helix. So they are wind, all of this polypeptide chain is wind on itself, and that is what gives us the tertiary structure. So now in case I have many of these tertiary structures, polypeptide, many of these polypeptides, okay, that are linked together by either ionic interactions, by either hydrogen bonds or other interactions together. That means many polypeptide chain. So you observe here you have one, this you have one, you have two, you have another one behind it, another one behind it, so 4. So in this case we call it a quaternary structure. So you note that it is made up of four polypeptide chains, okay, with each of them having their 3D conformation. So we can also group proteins based on their structure. So we can group them as either globular proteins or fibrous proteins. So what are globular proteins? Globular proteins, they are proteins that are very stable. They have a very stable tertiary structure. They are just like a globule. They are just like globe-like. So we can also group proteins based on their structure. We can group them either as globular proteins or fibrous proteins. So what are globular proteins? So globular proteins, they are proteins that are formed, that they are kind of spherical in shape. They have a very stable tertiary structure. They are soluble in water, and these proteins most of the time make up proteins like antibodies and hormones. So antibodies and hormones are globular proteins. Okay, so majority of these functional proteins are globular proteins. Majority of the structural proteins are fibrous proteins. Okay, so fibrous proteins, their secondary structure are more important. Okay, their tertiary structure, their secondary structure is more important than their tertiary structure. So they usually have an absent tertiary, meaning that what? Meaning that the polypeptide chains most of the time they don't wind on themselves to form a 3D conformation. Okay, so majority of these, they have just a secondary structure, and they make up insoluble. They are insoluble. They usually very tough, and usually perform structural roles, as we said. So proteins can become denatured when their 3D conformation is lost due to several factors such as heat, radiation, redox reactions, or reacting with heavy metals, or you could even add to this pH. So if the primary structure remains unaffected, you can always regain back the initial conformation of the protein, provided the conditions are good enough. So let's get into nucleic acids. So nucleic acids, okay, they are molecules that are essential for the formation of genetic material. So they are termed nucleic acid because they are found in the nucleus, okay, and they represent one of many milestones in biology because it has solved so many problems. That was the problem on how living organisms store information and pass it on across generations. So now we know that living organisms store their information as in some particular molecules that are found in the nucleus, which we call these molecules nucleic acid. And we also know, and we are going to see how these molecules can be transmitted to other generations. Okay, so the basic units of nucleic acid are nucleotide. Nucleotide, they are mainly made up of, so this is a nucleotide here. We can observe that the nucleotide are made up of a base, what we can call a nitrogenous base, okay, a pentose sugar. We have a pentose sugar, we call it pentose. You must have attended the class in Biomolecules 1, so you know what a pentose sugar is. Then a phosphate group, so they always have a base, a pentose sugar, and a phosphate. So these are the different nitrogenous bases that we have. We have two main types. We have pyrimidines and we have purines. What's the difference in between pyrimidines and purines? The main difference lies in the number of rings. So in pyrimidines, they are made up of just a single ring, okay, as you may observe just here. Whereas purines are made up of two rings, okay. So the different pyrimidines we have are cytosine. Then we have thymine and uracil, but now thymine and uracil is very important for you to note it that thymine and uracil are not found in every nucleic acid. So thymine is found only in DNA, uracil is found only in RNA. So both of them are pyrimidines having just a single ring in their structure. Then in purines we mainly have adenine and guanine. Okay, then different sugars we have ribose, okay, ribose, which is a pentose sugar having 1, 2, 3, 4, 5 carbon atoms. ribose, which is mainly found in RNA, and in DNA we mainly have the form in which this oxygen here at carbon number 2 has been removed. So we call it deoxyribose, and it is mainly found in DNA, as we said. And it is also important that you know that nucleosides are structures or molecules where you have just the base and the pentose sugar. Okay, when you have just the pentose sugar and the base, we call it a nucleoside. So for example, you might hear of adenosine, you might hear of guanosine, you might hear of cytidine, you might hear of thymidine, and others. So when you hear of this, you know that we are talking of a nucleoside which contains what? Which contains just the pentose sugar and the nitrogenous base without the phosphate groups. So condensation between the phosphate heads of a nucleotide and the sugar of another nucleotide leads to the formation of a dinucleotide. So this dinucleotide, so that condensation reaction leads to the release of water, and the bond that links the two nucleotides together is called the phosphodiester bond. It's called the phosphodiester bond. So it is not here, you should note. So multiple condensation reactions can lead to the formation of a polynucleotide. Okay, a polynucleotide which can form a DNA strand. So DNA is a double helix structure that is made up of two polynucleotide strands that run anti-parallel to each other. So you have two polynucleotide strands that are anti-parallel to each other, that are linked through hydrogen bonds that are formed between the base pairs. So it is important you know that adenosine pairs with thymine and uracil. Okay, so in DNA you always see adenosine paired with thymine. In RNA, adenosine is replaced by uracil, but they don't pair, since you know that, and you should know that RNA is single-stranded. Okay, so in everywhere where you see a base pair, two bases that pair together, adenine will always be paired to thymidine, to thymine, and guanine will be paired to cytosine. So AT pair is a double bond, while GC pair is a triple bond. So here we have to talk of two hydrogen bonds. Here we're talking of hydrogen bonds here, whereas here you have three hydrogen bonds. So it is not written here, but you should know that we are talking of hydrogen bonds and not the double bond which you normally know from your A-level chemistry. So the alteration in the different base pairs from the basis variation in genetic information. Okay, and we already said that the sugar in DNA lacks one O group, and that's why we call it deoxyribose. So RNA is single-stranded, as said, and it serves as genetic material for low organisms such as viruses and archaebacteria. So we have many types of RNA. We have messenger RNA. Their main function is to transport genetic information from the nucleus to the ribosomes into the cytoplasm for protein synthesis. So they permit us to transport that genetic information from the nucleus, okay, to the ribosome that is found in the cytoplasm, and the ribosome is going to assist in protein synthesis. Then there is transfer RNA or tRNA, which transports amino acids in the cytoplasm for synthesis of proteins. Then there is ribosomal RNA, which reads the message and is able to transform it into proteins. Okay. Then the different functions of nucleic acids are: 1. For the storage of genetic material necessary for proper functioning within the cell. They can help in the transmission of genetic information from one generation to the next. And they can also permit biological evolution of a species by altering its constitution in times of adversity. So here we have to talk of mutations. Okay, mutations that could be favorable in times of adversity. okay, because our genetic information is not fixed, it is variable, it can vary. There are errors that could be due to mutations, which we'll see that could be either positive, or it could either affect the species negatively. So in case it affects positively, it is going to help in evolution of the species. Then some other important nucleotides which you should know, we have adenosine triphosphate, that is ATP, which you know. ATP is a molecule that is energy rich and that our body uses as the energy currency, okay. It is used in the body during metabolism, and it is with ATP that our body is able to use energy, to transfer energy, and to store it temporarily, okay, to store energy temporarily for usage. So it is formed by combining the nucleoside adenosine with 3-phosphate. So as the name says, adenosine triphosphate, we saw what adenosine was. We said a nucleoside was a base where it was a molecule where you have just the base pair. The base pair, that's what here, the nitrogenous base, that's what that is adenine, that is coupled with the sugar, okay, with the pentose sugar, as the pentose sugar that forms a nucleoside. So we call it adenosine. Remember, we said everywhere you see O's, you should think of a sugar. So this ATP is formed by adenosine that is bound by 3-phosphate groups, okay. So the groups are termed respectively in ascending orders from alpha, beta to gamma, okay, and the third group, that's a gamma group, is the richest in energy. And the hydrolysis of this gamma group, the hydrolysis or the release of this gamma group, is the one that produces energy, okay. And we have ADP, water, we have ADP and a phosphate group that are released as byproduct, the usage of water. So it is associated with all metabolic reactions in the body involving energy. It is constantly recycled from its end products or precursors. So it is important during activities such as synthesis, okay, active transport, cell signaling, and locomotion. So other types of nucleotides, we actually call them coenzymes. So you have cyclic AMP. We are going to see this very deeply, especially when we do endocrinology. 3, we have nicotinamide adenine dinucleotide, okay. Nicotinamide adenine, that one is also abbreviated as NAD, which helps as a hydrogen transporter. It helps to transport hydrogen or to reduce some particular agents. So you have flavin adenine dinucleotide, or that is also called FAT, okay. So most of these are as precursors of ATP by transfer of a phosphate group onto the kinase. So for example, here you have the ATP. We said the ATP, you have mainly the adenine, that's a nitrogenous base, the adenine, you can observe it here. We said adenine is a purine, so this is adenine. Then we have the ribose sugar, that's a pentose sugar, okay. The pentose sugar, now at the level of this fifth carbon here, you have some phosphate groups that are added. So you have 1, 2, 3 phosphate groups that are added. So we call this adenosine. So these two is what we call adenosine. Then 1, 2, 3 phosphate, so triphosphate. Adenosine triphosphate. So let's look at this table that summarizes some very little important things which all of you should know. So here we are going to give, for each biomolecule, we give the monomer of the component, we give the polymer, the larger molecule, so the name of the polymer, and then we give the type of linkage that exists between the monomer unit. So carbohydrates, the monomer in carbohydrates are what? Monosaccharides. We saw the different types of monosaccharides. We had glucose, fructose, and galactose. Then polymers So the polymers of carbohydrates are called polysaccharides. Then the type of linkage are called glycosidic linkages. Then in lipids, the monomers are fatty acids and glycerol, and the polymer is called triacylglycerol, or we also saw it as triglyceride, so you can also call it triglyceride. Then the type of linkage are called ester linkage, so we saw it as ester linkage. Then in proteins, the monomers are amino acids. We said there are 20 of them. So you should know, you should check and do researches so as to know which are the essential amino acids, which of these amino acids are basic, which of them are acidic, okay, and which of them are essential. You should know of that. So the polymer for proteins are called polypeptide, and the type of linkages that exist in between amino acids in proteins are called peptide bonds. In nucleic acid, the monomers or components are called nucleotides. The polymer are the polynucleotides, okay, that form a strand. It could be an RNA strand or a DNA strand. Then the type of linkage that exists in between the nucleotides are called phosphodiester linkages. So that said, thank you for your kind attention and see you next.